Okay, we're going to get going today. We've changed the schedule, and the first speaker is Mukesh Verma, and he's going to be talking about epigenetics. And so in cancer, we learned it's not just DNA mutation. You can turn on and off the various genes. So he's chief of the Medicine Technology branch of the DCCPS. And Mukesh, welcome.
Thank you. So as he mentioned, I am in Division of Cancer Control and Population Sciences, and my program is called Epidemiology and Genomics Research Program. And I am a program director and branch chief there. So I will talk about epigenetics, and we are in Cancer Institute. So in cancer, first of all, this disease is very serious because as you can see how many people die with this, and new cases also come. In men, prostate cancer is the number one killer, and women, breast cancer is the one. And it is estimated, for example, in 2010, there were 13.6 million cancer survivors. By 2020, we expect 18 million. So we are making some progress, but at the same time, new cancers are also coming up. And in today's lecture, initially, I will tell you how the epigenetics came, so little bit background of genetics so that it helps you in understanding epigenetics. And then I will give you some examples how we can use this epigenetic approaches in clinic. And since we are epidemiology program, so we have a lot of cohorts and patient populations and cancer survivors, all that, how we can use it. Most of the cancers, breast, cervical, colon, prostate, lung, they take long time, like 14, 18, 20 years by the time they are fully developed. These are different stages of cancer development. So what is our idea that if we can detect that early, that is the best. So those markers are good, which can detect cancer early. And that gives us option for either developing some treatment or for stopping that. At the same time, and this is one example, that cancer is a multi-step process. So either by morphologically you can characterize or molecular biomarkers you can use. That way you can characterize, and based on what genes are expressed or what proteins are expressed, we make approaches for treatment. Now, few cancers are different, like for breast, cervical, colon, and prostate, you will see some stage will be from normal, it will be some precursor. But in pancreatic cancer and ovarian cancer, once it develops, then there are no markers for early detection, and right away you come to the cancer stage. So some cancers are such where mortality and incidence rates are almost the same. For other cancers, at least we have time to do something. Now about genetics, initially we did not know where genes are located at different chromosomes. So GWAS studies, which are genome-wide association studies, they identified different SNPs, those small nucleotide polymorphism at different chromosomal location, and that helped us that how functionally they are related or what is their location. So that helped not only in making genomic map, but association of these SNPs with the diseases. So that is the basis, and as you can see, most of the chromosomes have been very well characterized. Now about cancer, since genetics we know more, and in this case we know at least that in which organ sites, which mutations are there. So that information we have. Along with that, in cancer, mostly 12 pathways as proposed by Bert Vogelstein. Those are the major pathways which are related with cell survival or cell fate or genomic maintenance or instability. So that still stands. Initially, we characterized genetics, and other informations are also coming. And in epigenetics, which does not include any change in nucleotide sequence, but you will see different kind of advertisement even in magazines that happiness can come by epigenetics and all those, but that is not true, and I will give you correct scientific background. So epigenetics is stable alterations in gene expression by several mechanisms except nucleotide sequence changes. And that is the one, and we take advantage also. If you detect mutation, then at the most you can use for screening or follow-up of treatment. But with epigenetics, you can change those also. So you know about genetic code, but in epigenetics, two more codes are there. One is called methylation code, another histone code. Major difference in genetics and epigenetics is that genetics has static information, and that tells what you are capable of, what you can do. But epigenetics tells that what should be used and when it should be used and when should we stop. So it is in fact a regulator of genetic information. In popular magazines also, time to time, articles come about epigenetics. Here they emphasize that your DNA is not your destiny. Because as I will explain later on, that by changing lifestyle habits or environment, you can change your fate. So choice is yours. At the same time, at initial stages when people wanted to characterize or distinguish genetics from epigenetics, then they did some experiment in monozygotic twins. And these twins are such, genetically they are identical. But later on, they observed discordance in these twins and different, initially they observed for cardiovascular diseases, but later on other diseases were also found based on one part of the twin where they grew up and other one also. Same way, at the time of pregnancy, if diet also plays an important role, which twin will be developing what disease. So that kind of information we use for pharmacogenomics or pharmacoepigenomics, and microenvironment plays an important role. I mentioned about GWAS or genome-wide association studies, and now we are proposing epigenome-wide association studies. So little bit background about nucleosome structure. So it is made up of DNA and histones of H2A, H2B, H3, H4. In 2AB, in 2B, H3 and 4, most of the modifications are there. H1 is also there, but that is like a linker. And about 146 base pairs, they are wrapped around histone proteins. And the idea is that basic charge of nucleic acid should be neutralized so that your chromatin is stable. Otherwise, epigenetic regulation is at cell cycle or DNA damage or apoptosis at different stages. Environment, hormone, genetics, all those affect that. Different enzymes are for different processes. For example, for acetylation or deacetylation or methylation, different enzymes are there. And then recently, microRNA, they were also considered part of epigenetics. Initially, there was some controversial points. And in epigenetics, different kinds of modifications are there. That also in histones, that I will be describing. And recently, some non-histone proteins like polychromes, repressor proteins, they are also involved in that. So all these things work together, and methylation and histone modifications, those should be in equilibrium for stability. In methylation, DNA methyl transferase of different types, those have been characterized. Some transferases are needed just to initiate methylation, and others are needed for maintenance. In methylation, when I say, then cytosine at 5 position, that gets methylated. In histones, acetylation, methylation, phosphorylation, ubiquitination or biotinylation, all those modifications are found in histones. And long or small non-coding RNA, they are also part of that. And in genomic imprinting, especially for IGF gene, that has been also characterized that it is regulated epigenetically. About methylation, in promoter region, CG sequences, if they are more than 60% in a 0.4 kb range, then it is called CpG iodine. And if CpG that gets methylated, then it results in inactivation of gene, and process is called hypermethylation. And this has been reported for tumor suppressor gene. Similarly, sometimes promoters are already methylated, but they undergo hypomethylation. So they lose methyl group, and that has been observed in oncogenes. So both ways methylation affect cancer development. As you know, tumor suppressor genes, they will try to suppress tumor, while oncogenes try to increase that. I mentioned that age, diet, disease, lifestyle factor, they play a major role in epigenome. And in genome, as you know, genome is sequenced only once, because no matter from which cells you take DNA, main DNA is the same. But if you want to study epigenome, then you have to sequence or characterize many times. At the same time, in twins, when different diseases were studied, then it was found that those diseases which are neurological disorders or few selected diseases, in that genetics plays a major role. But in diseases like cancer, it is epigenetics which has a dominating role. So more and more data is coming. Throughout your life cycles, epigenetic changes happen. And we have at NIH one epigenome roadmap program, which we initiated on the same pattern like whole genome was sequenced from, and in epigenome we wanted that pathologically normal people, if their histone profiling or methylation profiling or microRNA profiling if we have, then they will serve as reference to compare with any disease samples. So when it was done at initially stages, when cells are only 2 or 4 or 8 or 16, 32, 20 different kinds of cells were characterized epigenetically, and then it was found that quite up to late stage there were no mutations or SNPs, but epigenetic changes are always there. Here maternal factors are more important, or environment at this stage. Later on, lifestyle and diet and other things come, but throughout the life, epigenetic regulation is there. And as I mentioned, that our program is interested in understanding epidemiology, where we want to identify who is at high risk of developing cancer. But after development of cancer, we treat them and follow for secondary cancers or cancer recurrence or elongation of survival. So we follow in our studies almost all stages of life, and we keep samples. And these are some of the examples where you can take samples, and these samples can be characterized epigenetically. And since lifestyle plays a major role, you can imagine if a pregnant mother is there, then 3 generations are affected. First her own life, then baby's life, then stem cells are there, where also epigenetic regulation is there. And in some cancers, at least for example in lung cancer, smoking or tobacco is a direct causative agent. It is similar to HPV or papillomavirus is a direct cause of cervical cancer. So at least in those cases, if we can avoid that, then cancer will be reduced. Now in environment, many toxic substances, they are regulated epigenetically. For example, arsenic, genetic epigenetic regulation, benzene by methylation, cadmium, and these are there. And in fact in arsenic, that gets mono or dimethylated also, so it will deplete whatever methylation we are supplying. So most of these environmental agents, they are regulated epigenetically. And few of them are such, they are not mutagen, but still you see cancer. So that's how epigenetics come into picture. Another point I want to make, that whenever some exposure, radiation or some other kind of environmental insult is there, then first thing affected are transcription factor or cell cycle regulator. If it is for little longer time, then epigenetic markers, they are affected. When it is very, very long time, then only mutation or other things come. That also emphasizes that if we characterize these steps, then it is better than to go for genetics. Genetics also helps, but as early we can check that, that will be good. In this case, they have presented about how many translocations, deletions, amplification, etc. in different cancers they have observed. Now in epigenetics, first changes are seen in histone modification. So histone gets ethylated first, and enzymes which are involved in that, they get inactivated, and then it recruits methyltransferase and methylation of DNA happens. And if DNMT1, 2, 3, all those are involved, then it is stably silenced. Peter Jones and Andrew Feinberg, they are the pioneer 30 years ago. They proposed this idea of epigenetics regulation of genes. And now chromatin, if you see top part of the figure as well as bottom part, this is much more compact chromatin, this is relaxed. So here transcription factors can go in or out, so gene is active. But gene gets inactive as chromatin comes closer. Red color represents hypermethylation, yellow color represents deacetylation. And many other steps happen, but it is a gradual process by which gene inactivation or expression of gene happens. And methylation also, one by one, these CG sites get methylated. So if you are doing some sequencing experiment, then both strands you should sequence. That's the whole idea in this picture. So deoxycytidine is converted into 5-methyl deoxycytidine. Throughout life, global hypomethylation is observed. In gene or gene nearby gene like promoter region, you will see more CG sequences or methylated. But in line, sign, and ALU, all those sequences, they already have lot of methylation in that, and with age or time, they get hypomethylated. At the same time, promoter get hypermethylated, and histone modifications with age that increases. That is almost universally present. And especially these line or sign sequences, they are hypomethylation that is involved in stability of genome. And if we study methylation little in detail, then it can be abnormal increase, abnormal decrease, or no change. In tumor suppressor genes, as I mentioned, they get inactivated. So either methylation of both allele, or one allele methylation and another mutated, or imprinting of the gene. That can happen in abnormal decrease, like proto-oncogenes, they are affected, or in early 90s, gene therapy came where retroviruses people use to introduce new genes into that. But many times gene therapy did not work. At that time, epigenetics was not characterized well, but now we know that promoter of those retroviruses, which are called LTR, they get inactivated by this mechanism. Or another is increased DNA rearrangement and possibly aneuploidy. When no change, that either chemically induced methylation or poor repair or deamination. So these are different mechanisms in methylation which work. And now, since the technologies are advanced and much more research has been done, then whole pathway also have been characterized where only those genes people can select which are regulated epigenetically. So here integrin signaling is shown. Now microRNAs, 10-12 years ago, people thought that they are only in animals or only in plants or Drosophila, but later on they were reported in human. And now we know that not only human but organ-specific microRNAs are also there. So we want to utilize their profiling in different cancers or polymorphism in the coding gene of microRNA. So that is a big field. And since microRNA are of a smaller size, like 21 nucleotides or so, and because of their stem loop structure, they are very much stable. When people try to do expression analysis using mRNA, mRNA is much more unstable, but microRNAs are stable. So this is one of the examples that how microRNAs are involved in metastasis. Coming to histone, most significant histones in cancer are S2A, 2B, S3, and H4. And these are the modifications in some or the other cancer or other diseases. These modifications have been shown. So I was mentioning about NIH Epigenome Roadmap program. Initial work in epigenetics was done on methylation because that was based on DNA analysis. Histone work could not be completed because monoclonal antibodies against these modifications were not available. But through Epigenome Roadmap, we gave contracts to different companies. They generated monoclonal antibodies which are of high quality, and then they characterized that which modifications are involved in cancer. So that work is also going on. So now some specific inhibitors also have been identified based on these specific sites. And techniques are also such that at different locations we can follow up also how much methylation is there. So relative quantitation also you can. Same thing is for methylation as well. Since I have been in this field, so initially on epigenetics and cancer prevention for New York Academy of Sciences, I edited this book. And then in 2012, another book on cancer epigenetics. And since we do epidemiology, and there we want to incorporate all that information, so some factors are those susceptibility factors, like immunity or your genetic background, how those are involved in epidemiology. This one I edited, and another one on modifiable factors like lifestyle, energy balance, alcohol, tobacco, all that. So all these things help us in understanding epigenetics more. And this year, this book has come where we have emphasized that what kind of samples we can use, which can be collected non-invasively. And we can do early diagnosis, or after treatment we can follow up. I was interviewed by Nature, and then about epigenetics discussion was there. So there I emphasized that epigenetic changes are reversible and therefore have edge over genetics. And as I was mentioning, in genetics you can detect translocation or deletion or insertion, SNPs or mutation, but you cannot put it back. But those modifications which I showed, like microRNA profiling or methylation changes or histone changes, those you can change by chemicals. So another time also I was interviewed by Nature, and there I mentioned that successful approval of first generation of drugs intended to target epigenetic pathway has convinced almost every major drug company to invest in cancer epigenetics. So to date, 4 compounds have been approved by FDA. At NCI, we have some trials going on, and major pharmaceutical companies, they are focusing on that. And if some of you go to ASCO, American Society of Clinical Oncology, then every year they update. And I also keep track of that. So that is the major potential and people's interest in epigenetics. And in almost in all diseases now, regulation has been observed. I am discussing about cancer more today, but in neurological disorder or many other diseases or psychology, you don't have much phenotypic characteristic. In cancer, you can see tumor and follow-up. In those cases, epigenetics is being utilized more. And when you plan an experiment in methylation, so to reduce false negative or false positive, you have to keep an idea about methylation content or level or methylation pattern or level profile or pattern profile, all these things. And these have been very well characterized. So if you keep in mind that, then you can get quantitative results. Steve Balin, who is in Johns Hopkins, he is characterizing those proteins which bind to promoter or near transcription factors, and they regulate. So in euchromatin stage or heterochromatin stage, what is the sequence of different proteins binding to that? Some are methylated DNA binding proteins, then histone acetyltransferase, histone deacetylase, all those come. So he has characterized very well. Along with that, in a lung cancer treatment, some trials he has done very good. And initially he showed that some tumors are there, but by treatment, he could treat those. When we collect samples, then during different stages of cancer development, exfoliated cells are also important because they are shed many times and you can collect and use. And in last few years, you must have heard about exosomes or microvesicles. They are also released from tumors. Tumors as well as normal, but tumors they also contain some microRNA, mRNA, lipid. So those are also being used in diagnosis or prognosis. Some of the well-characterized group of genes in different cancers that I have shown. And so in several reviews also I have covered those. And now in this figure I have shown these are different categories like histone deacetylase or serotinins or histone DNA methyltransferase or histone acetylate. So these are different steps, and those genes where these proteins or enzymes they work, those are shown here. So these have been characterized more and more. The idea is, which mostly pharmaceutical companies and some researchers are doing, that they are targeting these proteins and then seeing how much this gene is affected. So that work is going. And clinically also, for different compounds I have shown, some are already, as I mentioned, 4 are FDA approved, but others are also coming up at different stages. These are, he is one of the person, Jean-Pierre Issa, who is in UPenn in Philadelphia. So he has developed that, and now Zibulerin or Saha, these are very good compounds which are giving good results, although in some cases you will see some kind of side effect which with any new drug can happen, but they have good potential. Now, for different enzymes or proteins, I have listed those name of the drugs which are at different stages of development. So that is also there. So a lot of things are going on. And these inhibitors which are being characterized or made, they are either short-chain fatty acids, aliphatic compounds, or cyclic tetrapeptides, or benzamides, or different things. And histone deacetylase also, initially only 1 or 2 kinds were there. Now more characterization have shown that number 1, 2, 3, and 8 are also there. So companies are targeting different forms and trying to study in which cancer, which histone form is more prominently expressed. So these inhibitors are providing very new class of anti-cancer drugs because they target cell cycle arrest, differentiation, migration, or apoptosis. And sometimes few SDAC inhibitors act as radiation sensitizer drugs, resulting in better radiation therapy. So with that, what I mean, that if you treat with these inhibitors first and then do radiation, like for cancer treatment, mostly it is by radiation, by chemical treatment, or by other changes. So that way also they have. Now I will give you 2 examples in which in combination of these demethylating agents and histone methylation agents they work. So in this case, 5-azacitidine, which is methylation inhibitor, and valproic acid, which is deacetylating, these were used in combination, and patients were such in which no other treatment worked. So in those cases, consent is taken and whatever promising is coming, that is tried. So in these 55 people, it was tried, and 28 day cycles, both were given in combination. And now technologies are such that we can collect blood and analyze for histone or methylation at different stages, and they started giving response. So that is one example. In another case, it was combination of 5-aza, valproic acid, and transretinoic acid. In this case also, more than 50 patients were such which did not respond to other treatments, but they responded to these one. And few reports are also coming like PARP inhibitor will work better if you include either methyl inhibitors or histone deacetylase. At NCI, we have some clinical trials. In this, histone inhibitors were used, and this website I have mentioned, but in if you type cancer.gov or you are at NCI website and go under clinical trials, and in that if you put histone inhibitors or methylation inhibitors, you will see how many trials are going on. And these are not only for solid tumors, but different kinds like lymphoma or breast cancer or glioblastoma. In different kind of cancer, these kinds of inhibitors, they have been working. At the same time, methylation inhibitors also in different cancers they are doing, and in 51 studies, they are being used. I don't want to say that it has resolved all those problems because whenever epigenetic changes are there, they are in all cells. But at least in few cases where that has been characterized more, that is working. And here, another thing I want to emphasize that you will hear about personalized medicine or specific medicine, like each person is different. Some drug will work on one person but will not work on another. In few cases, we know that genetic mutations, they determine response. But now more and more reports are coming that your epigenomic profiling also contribute to response to a drug. So this kind of information is for that also. And Bristol Myers, Schellingprow, or AstraZeneca, Merck, all these companies are heavily involved in these inhibitors. So the idea I want to give you that environment and endogenous hits, they damage RNA interference or chromatin or DNA methylation, but they also affect genetic changes, and both are interlinked. So whether gene transcription, repair, or cell cycle checkpoint are there, they are affected, and other steps are also affected by other mechanisms. So in whole cancer development, genetics and epigenetics, they go side by side. And these are those 4 inhibitors which have been approved by FDA. 2 are for methylation, 2 are for histone deacetylation. And in one example here, they have shown for lung cancer they have been used and they were successful. Another example is for hematological and epithelial tumor cells. Now I will give you a few examples in different cancer. In colorectal cancer, those genes which are affected or regulated epigenetically, they are shown here. They are expressed either early or late, and a lot of work has been done on colorectal cancer. And in fact, in cancer, left side of colon behave differently than right side of cancer. So all these profiling is different. And also one profiling is called SIMP profiling, CpG Ireland methylator phenotype. So that was unique for colorectal initially, later on for pancreatic and ovarian also that has been reported. So in this study, basically several samples were taken together and then integration of genetics and epigenetics was observed. For example, KRAS mutation or BRAF mutation, and then microsatellite instability, whether it is high or low, that was combined with methylation data, and based on that combination, some bioinformatics was applied, and based on that, patients were identified where either one modification is there or two are there, or if all three combined, then if someone has combination of modification of these genes where MS, microsatellite instability and some mutation and epigenetic changes are there, they were at much higher risk compared to others. So multiplexing of markers is also coming up. In colon cancer is the one which is the most characterized tumor in terms of genetics and epigenetics. In ALL and AML, just based on methylation profiling, different populations could be separated. Similarly, in lung cancer also some genes have been characterized, and in this case, Steve Belinsky, he has identified some methods like from sputum, non-invasively he can collect and then characterize annexin 1 and 2, these are proteins, and then methylation markers, if they combine them, then the sensitivity and specificity of their assays improve, and we can detect lung cancer early. Mesothelioma, this is also a type of lung cancer which due to asbestos exposure that happens, and in that case is also what are the genes which are regulated by methylation, those have been characterized. In esophageal cancer, where Barrett is the precursor, in that also 20 genes at a time they were characterized, and you can see wherever gray or dark color is there, these are level of methylation. So from the same person, in normal stages or after development or disease, these samples were taken and a gradual increase in methylation was observed. So once you enroll some patient or some of those people who are living in high risk exposure, we collect the samples in our cohort. 34 of those cohorts we maintain in our program, and they come after 6 months or 1 year or 2 years, and we collect samples. So suppose we get a marker today, then we want to see when these marks appear first, then we can go back and retrieve those samples retrospectively and then see when we saw the marker or change first. So that helps us in developing new markers. And methylation profiling has been correlated with survival also. In pancreatic cancer, as I mentioned, we don't have early tests, but whenever people are enrolled in some trials or cohort, then we take all that history of the patient or lifestyle or diet or exposure. So those things help. For example, if somebody is alcoholic, long-standing diabetes is there, KRAS mutations are there, and then P16 and P14 are hypermethylated, then a person is at high risk of pancreatic cancer. So that's how clinical information is used. After the treatment also, methylation profiling can be used whether treatment is working or not. Infectious agents, in about 15% of cancers, infectious agents are involved. Whether it is HPV for cervical cancer, or hepatitis C, B for liver cancer, or for HHV8 and others for Kaposi sarcoma, or H. pylori for gastric cancer, all those are there. So these, or EBV for nasopharyngeal carcinoma, they are there, and in that epigenetically latency associated nuclear antigen or Epstein Barr virus nuclear antigens, those were the main proteins or those genes coding those were involved in development of cancer. Because many times these viruses or infectious agents will be in the body and they will be for a long time. In many cases, we don't know what triggers them when they start expressing. So these kinds of mechanisms are helping us, and that is the reason why we do such kind of a study, that asymptomatic healthy carriers, those are there, but we don't know when they will develop the disease. But if we have profiling in these cases, then we can predict that who is likely to develop cancer. And recently, or a few years ago, about HPV, EBV, and HBV, complete methylome has been done. And here it is shown that whether early genes or late genes or other genes in virus, when, which got methylated, how much it got methylated. So all those profiling as a reference point, they have been collected, and that has been very helpful in identifying SYN1, SYN2, and SYN3 different stages. So for molecular classification also, this information is very useful, because otherwise only pathologists, when they tell some disease is there, then only they can distinguish. But many things pathologists cannot tell. So this molecular profiling is important. In hepatocellular carcinoma, although several other genes have been characterized, but sometimes we find such genes which are only for some population. So population-specific genes are also there. And in the US, as you know, people come from different countries. So our focus is that whatever we develop, that should be applicable to different kind of populations. And these are other genes which are affected in hepatocellular carcinoma or liver cancer. In oral cancer also, these are the group of genes which are regulated by methylation. In immunology, if we go, then B cell activation and C cell activation, T cell activation, different steps are regulated epigenetically. So that way also some basic research is going. For prostate cancer, PSA has been used, but if levels are below 4 nanogram per ml, then doctor cannot say whether prostate cancer will be developing or not developing. So in those cases, GST-P1 methylation has been found very good. And in most of those cases, these markers appear much early than others one. And initially, some kits were developed based on these GST-P1. And then Quest Diagnostics, they have acquired this test, and this for prostate cancer, that is being used. And PSA is good if level is 15 or 20 or higher. Then easily you can do, but just to make a decision whether treatment should be done by surgery, radiation, or chemotherapy, for that these methylation markers are important. And these markers from urine also you can collect exfoliated cells and characterize by methylation. This example is for bladder cancer, where in urine, this gene was characterized. And then in the same sample, if other cancers are coming up or they are at different stages, because many times if one cancer develops, then secondary cancer or multiple cancers also come time to time. But which one will come first or which one has more chances? For example, if somebody is a smoker, then lung cancer usually comes first, then bladder cancer appears. So those kinds of correlation also by methylation profiling you can know. And Peter Laird, who is in University of Southern California, he has done that kind of work. Now, in cancer field, about 30 to 40% of cancers you can either suppress or prevent by changing lifestyle or diet. So that is there. So we have to pay more attention about that, and I will give you a few examples where natural food or their byproduct of those foods, they are present and they have properties of demethylating enzymes or deacetylating. In this case, just by changing methyl donor without any exogenous carcinogen, such situations were created where models were created for liver cancer. So just by changing diet, that was shown. And in this slide, I have shown that some certain compounds like tomato, apple, or citrus fruits, they have DNA methylation properties. Some have, like cashew or grapes or garlic, they have histone modification, but some certain foods or natural food compounds are such which have these properties. And Institute of Alternative Medicine and in many other institutes also, they are trying how we can utilize that information more and more. Like FDA will approve only a specific compound, but at the same time, in combination, if you use natural food and your habits, then how we can do that. Now a little bit about NIH roadmap. As I mentioned, this was created by Dr. Zaharouni when he was NIH director. Idea was that such mechanism, which is involved in many diseases, many institutes are involved, there was no such type of system where you can study and we can put a lot of money. And at the time, genome sequence we completed, but epigenome was not done. So 2 projects were started. One was about microbiome, another was about epigenetics. So in that case, why it was done? Because we wanted such program which takes 5 to 10 years, big projects. Individual investigators cannot do that because those kinds of projects are not funded easily. And by this we wanted to accelerate pace of discovery, and this should be transformative. And as I discussed about those informative, so those kinds of conditions were completely filling with that, and it should be trans-NCI. And now on the same pattern, Common Fund has also come. And I am involved in Common Fund metabolomics as well as microbiome also. So when it was approved, so idea was that total this much money initially we committed from 2008 through 15. But now we are at this stage and we are expanding that also. But whole idea was that first of all we wanted to map those sites like methylation sites, histone profiling, microRNA profiling, and chromatin compactation by DNA is one analysis. At the same time, some projects were disease specific, like suppose NCI wants to take on cancer, we can do some research on cancer, or NIDDK on kidney diseases they can do. And one center we made data analysis or data coordination center. In genomics some data come, and by this program or other program like ENCODE or TCGA, huge amount of data will come, like in proteomics it comes here. In genomics you can see it is of one order, but if you include methylation or histone modification or microRNA, so to keep that also we need more space or more capacity. So NCBI and other people kept, and last month we gave contract to 4 outside NIH agencies, and we are renting clouds so that Broad Institute, MIT, and University of California, San Diego, and Santa Cruz, they will be maintaining all that data. Whole idea is that whatever data comes through this one or Common Fund like metabolomics data is also coming, they will keep as well as develop tools, and investigators in the lab in the university or NIH, they can use that data and then try to utilize or integrate genomic or metabolomic or epigenomic with other information to get better results. So that was the reason it was developed. After we developed, then Europeans started IHEC program, International Human Epigenome Consortia. In this I represent NCI or NIH. For roadmap I represent NCI, but in this program I represent NIH. Hello. So not to overlap with the roadmap, they emphasize that in cell line or other model system also they should do epigenome profiling. And with aging also they wanted to do, so they are doing separately. And when it was started, then so many countries got involved. Like human epigenome is only US, but these people are also working on human in Europe. And funding agencies are also many, including Wellcome Trust. And industry, Epigenetics, Novartis, they are also putting money. So huge amount and lot of work they have been doing. If in Google you put IHEC or any program you want to know more about that you can do. And when it was initiated, at that time Nature and Science covered this program. And this is their website. So overall I gave you the view, and in epidemiology how we want to utilize this epigenetic information, whether these markers can help us in identifying new risk factors, or how they can help in identifying those populations where disease have not developed but they are at risk, or after disease is developed, how to treat them, and whether we can prolong their survival or not. In cohort study it is more important because their disease has not developed and we follow them. How much they are correlated, genetic and epigenetics. Whether some race or ethnicity is specific, these marks are there, especially microRNA. And then how we can use that for better defining the subcategories. For example, in breast cancers, maybe 20 kinds of cancers you will see. So these molecular markers will work, and these are some of our key investigators. These are my collaborators, and I will be happy to take questions. Thanks a lot.
So I guess with this you're able to get information on methylation quite readily.
Yes.
What about acetylation?
Acetylation, that is also coming up now. like RNA-seq came. Now for histone also, lot of companies can do. They are at this stage in individual investigators' lab. They can do, but high throughput is not there. So the way it is working, when people submit project, we say, okay, make a subcontract for that company because for the time being, that is established. So to understand basic mechanism, they can use our monoclonals and can do that. But if they want to do epidemiology or large number of samples or in trial, then they can contact company. And same approach we are doing for metabolomics also, like Metabolome company and others. They can do NMR or mass spectrometry on thousands of samples. So both ways we are. Initially, we thought everything should be done in our lab. Then we realized whatever industry or biotech companies are developing, we should integrate. But that is coming up very, very promising. And if later on also, if you have any question on global, you can find my email or other things. So I will be happy to discuss with you. Or for your future also, if you want to discuss, so those things are also fine. Okay, thanks a lot.
So unfortunately, Dr. Zudaire could not be here today to present, but through the miracle of modern technology, we'll show one of his lectures from last year.
Thank you, Terry. Thank you so much. So yeah, my name is Enrique Zudaire. I'm at the Tumor Angiogenesis section. And I'm going to be talking mostly about tumor angiogenesis for the next, hopefully, 45 minutes. Thank you for staying here. It's very cold outside, so you might as well enjoy the warmth of the room. So I would like to begin to my presentation by defining what angiogenesis is. And I use, often this slide that compares two processes that are related but that are different. One is angiogenesis, and the other one is vasculogenesis. So angiogenesis is the formation of vasculature from pre-existing vessels. And I have a little video in here which demonstrate that. This is from a publication, some studies done in zebrafish. Here you can see a vessel, and you can see how additional vessels actually grow during embryo development of the fish. So this is what we call angiogenesis. A different process is vasculogenesis, which is also the formation of vasculature, but in this case from undifferentiated angioblasts. And both of these processes are very important, very prominent during embryo development. Angiogenesis also occurs in adult organisms. There are scarce reports of vasculogenesis occurring also in adults, but it's not as prominent as angiogenesis is. So formation of vasculature from pre-existing vessels. So angiogenesis is, I like always to talk about, physiological and pathological angiogenesis. And obviously angiogenesis, I don't know if this is obvious for you or not, but angiogenesis is a very important process physiologically speaking. Not only during embryo development, but through a number of different processes, some of which are detailed here. Female reproduction system, corpus lutein formation, wound healing, for instance, extremely important for wound healing. Wound healing is a very angiogenesis-dependent process. And then we also have pathological angiogenesis. So as we're going to talk about later, angiogenesis is a very tightly regulated process. And when this process is deregulated or becomes deregulated, it is normally associated with a disease state. And so we can have too much angiogenesis or we can have too little angiogenesis, and that in both of those become pathological situations. So in some cases, we want to stimulate angiogenesis. For instance, we have a heart attack, and in the recovery of the heart attack or in a wound healing, for instance, we want to induce angiogenesis since these processes are angiogenesis dependent. In other cases, we have too much angiogenesis, and the therapeutic goal will be to be able to block angiogenesis. So there is a number of these diseases, like for instance, hemangioma, psoriasis, and most notably tumor growth and metastases, which are related with angiogenesis. And that, as we're going to see later, there's in 1971, actually, Judah Folkman put forward a hypothesis that if we were able to block angiogenesis, to control angiogenesis, we'll be able to retard or delayed tumor growth. This is a very simplified story of tumor progression, okay? Here we have our happy normal cell that, for reasons that we're not going to discuss now, becomes a tumor cell. This tumor cell is fully tumorigenic. In the right context, it could become a clinically relevant tumor. However, it needs to go through a series of steps. During the initiation and promotion, this cell divides, and it reaches a certain size. That size of a tumor is what we call dormant in situ cancer, dormant tumors. I like to cite this seminal publication from Black and Welch in 1993. And actually, the focus of the publication was not what I'm going to describe now, but it's mentioned in that publication. These two doctors that were looking at trauma patients for small lesions, malignant lesions. And what they found was, I think, surprising, and is that, for instance, age 40 to 50, 39% of women present these lesions in the breast. Age 60 to 70, 46% of males present these lesions in the prostate. However, etc. However, only about 1% of these lesions are ever diagnosed, meaning 1% of these people go to the doctor saying, I have a cancer. Okay? So what this means is that, all these 38% remaining, those cancers, they never developed. Those cancers are there, but they actually never evolve into a clinically relevant entity, which I guess is a good thing. So these numbers, that there is now a number of publications that have supported the same observation, and these numbers are actually going down, and these numbers are going up, meaning this is a normal thing. We all have these things, at least when we reach a certain age. So there is then a data to support these dormant in situ cancers, which are again fully tumorigenic cells that could develop into tumors, but they don't. Actually, there are some interesting studies that they take these lesions and they put them in a different environment, and then these lesions are able to grow into clinically relevant tumors. So what does these tumor mass need in order to grow into a tumor? Well, there is a number of characteristics within the tumor microenvironment in these particular lesions that allow those cells to be able to recruit vessels to induce angiogenesis, what we call angiogenesis. So there are a number of different stimulus that can occur in the tumor microenvironment. One of them is hypoxia. These cells, when they reach a certain size, because they are not vascularized, there is no oxygen reaching these cells, they become hypoxic. And hypoxia is a powerful stimulus for angiogenesis. So there is a crosstalk that occurs between the tumor cells and the endothelial cells in nearby vessels, which results in induction of angiogenesis and a sprouting of vessels from these vessels toward the tumor cells. So this is how angiogenesis occurs, and it's at that point, once the tumor gets vascularized, that it can grow exponentially and reach a clinically relevant size and metastasize. I want to emphasize, I just mentioned tumor microenvironment. So this is not only a game between the tumor cells and the endothelial cells within the tumor microenvironment. There is a number of different cell types and both cellular and extracellular elements that participate during the angiogenesis process, and that are going to be important. And I think I'm going to be mentioning a little more about this in a minute. So, as I said before, angiogenesis is very tightly regulated. Under normal conditions, there are stimulators of angiogenesis and inhibitors of angiogenesis that are balanced. And angiogenesis is controlled. For instance, my 4-year-old daughter, in the summer, she has constant scabs in her knees, right? So it's like a constant wound. So thank you for angiogenesis. It's this regulated process that allows, these knees to heal constantly during the summer. So perfect balance between these stimulators and inhibitors of angiogenesis. Those in situ tumors that we were talking about before that don't develop, something like that occurs as well. There is kind of like a balance between pro-angiogenic and anti-angiogenic factors, and the tumor stays at that particular size. If there is an imbalance in these factors, either too much stimulators or too few inhibitors, then is when angiogenesis occurs, and then is within the tumor microenvironment when these cells, can grow to clinically relevant tumor. I like this slide because it also mentions some of the other players within the tumor microenvironment. There is a whole slug of inflammatory cells which are recruited to the tumor site and that participate in the tumor angiogenesis process. There is endothelium, of course, from the vessels. There is fibroblast. There are T cells, stroma. So there is a lot of different components within the tumor microenvironment which are important, not just the tumor cells or the endothelial cells. And this is just a representation of that idea that a study that we did a number of years ago, where we looked at the influence of certain inflammatory cells within the tumor microenvironment. So this is summarized basically in this slide, where we have the tumor cells, which, because of the distance from these vessels over here, they are in a hypoxic environment. And that induces, or that allows the tumor cells to produce and secrete a number of factors which are angiogenesis stimulators. These angiogenesis stimulators create a gradient towards this pre-existing vessel and induce the migration of inflammatory cells to the tumor microenvironment, where they can actually release additional factors, which are pro-angiogenic factors, and then is when this angiogenesis process is enhanced, not only by the tumor cells, but all of these components within the tumor microenvironment. These vessels that reach the tumor, these tumor vessels basically, are different than the normal vessels. And this is, for instance, a case. I wanted to show this because it illustrates very well, how the vasculature invades the tumor. This is a case of a 14-year-old with a large pheochromocytoma right here. This is a kidney, backbone, ribs, and the pheochromocytoma is here, which I have digitally extracted so you can look at the vessels. And you can see how all these vessels are invading. All this is neovessels, neovasculature. That vasculature was not there before. So it's grown towards the tumor, it's invading the tumor, which in this case is very involved. And but one of the things that you already see here is not only that the tumor has a lot of vessels, but that these vessels look different than the normal vessels, like these vessels over here. So one of the things that you can observe already is that they are very tortuous. They're not straight, nice straight vessels, but they're very tortuous vessels. So this is something that is being studied, and indeed, normal vasculature, normal vessels, they have a nice parasite coverage. I'm not sure you're familiar with these cells, parasites, but parasites cover vessels, cover the endothelial cells in the vessels, and stabilize vessels. They have, for instance, a reduced integrin expression. They are very tight. Normal vessels are not leaky. We're not losing blood, all over our body. They're really tight vessels. And this is in contrast to tumor vessels, which are very tortuous. The morphology of those vessels is different. They've lost pericyte coverage, and therefore they are very leaky. Tumor cells are very leaky. Interestingly, and this is something that I'll talk about later, they also express markers that are not expressed in the normal vasculature. And this is what they're called tumor endothelial markers, and is the main subject of study in our group. But you can already understand that if these vessels, these tumor endothelial cells, express tumor endothelial markers, meaning markers that are not expressed in the tumor vasculature, we have a mean to differentiate those vessels, and we have a way to develop therapeutics that are able to target specifically those vessels. Just an example of how these vessels look like. Here, for instance, you have normal vessels, and here you have tumor vessels already, very easy to differentiate just by the morphology of these vessels. Another example using a scanning electron microscope, striking differences between the normal vessels over here and the tumor vessels, and just representation of how leaky these vessels are. Some of these tumors actually are full with blood. They form these blood lakes, and it's just because the vessels are extremely leaky and they continuously, leak blood to the extracellular medium. So based on this, as I mentioned before, Judah Folkman in 1971 in this paper in New England Journal of Medicine proposed that if two things. One, that tumor growth is dependent on angiogenesis, and two, if we can actually therapeutically target this vasculature, we'll be able to delay tumor growth. So this happened in 1971, so it's been like 42 years until today. So there's been, we're pretty much going to skip those 42 years, but I'm going to tell you that yes, there's been a lot of efforts to develop anti-angiogenic therapeutics, and is one of the most promising actually fields in oncology. And so can we cure cancer with anti-angiogenic therapy? So I guess the answer is yes, and this was demonstrated in as early as 1997. And in this publication in Judah Folkman's lab, what they show in different tumors is with this particular anti-angiogenic compound, they can grow tumors. And when they start administering the anti-angiogenic compound, the tumor regresses. When they stop adding administering the anti-angiogenic compound, the tumor starts growing again. They administer it back, and they can go cycles, of giving the anti-angiogenic compound or giving some vacation period, basically between the two. And you can see how the tumor growth goes up and down. And they were able to show this in a lot of different tumor types, demonstrating that the tumor growth is very much dependent on angiogenesis. And if you block angiogenesis, basically you are able to block tumor growth and regress the tumor. Now, these are xenograft models. These are mice. And when people asked Judah Folkman, can you cure cancer with anti-angiogenic therapy? And he said, sure, in mice. I can cure them in mice. A completely different story is what happens in humans, right? And when we try to translate all this into the clinic. And this is one of the things that I want to talk about, and I have a number of slides that will illustrate, the problems or the challenges, basically, of translating this particular anti-angiogenic therapeutics into the clinic. As a matter of fact, this one, endostatin, is one of the therapeutics that fail, basically, in the clinic, even though results in preclinical models were so striking. Yeah?
What is the wide base on this?
Oh, this is, you have to read the paper. This is how, basically, they've done the studies in so this continuous administration of the drug at that point. Okay, so as I mentioned before, there's been a tremendous effort to develop therapeutics targeting pro-angiogenic compounds. And this is just a summary, temporal line, basically, of all those, anti-angiogenic therapeutics that have been targeted. And it's not comprehensive. There are more than the ones that are shown in here. And these therapeutics actually cover a whole spectrum of different molecules and factors within the cell, addition molecules, endogenous inhibitors, angiogenic factors, protease inhibitors. So there's a lot basically of different types of factors that can be targeted and that have an anti-angiogenic effect. And it's also worth mentioning that Verma is quite interested in this field, and so they invest heavily in anti-angiogenic research, which is a good thing, actually, for the patient eventually. So there's a number of different pathways, as I was saying in the previous slide, that can be targeted in order to target angiogenesis. One of the things that I haven't mentioned in the interest of time is that angiogenesis is quite a complex process that can be seen as a orderly series of steps that are required basically to, from the moment where endothelial cells get activated, they start proliferating, they need to move around within the tumor microenvironment, they need to align themselves, interact with each other to form a tube. So it's quite a complex process. And from the perspective of people like me that try to block this process, all of those steps represent an opportunity to block angiogenesis. All of those steps are required, and therefore all of them, are an opportunity to block angiogenesis. And therefore, there's a lot of different targets that can be used. This is a summary of FDA-approved anti-angiogenic therapeutics. And let me see. Yeah, it's actually not up to date, but it's a pretty good summary. And the one thing that I want to highlight here is that even though there is a lot of different pathways that can be targeted, most of the efforts, today have been focused on this particular molecule in here, which is called vascular endothelial growth factor. It's one of the earliest pro-angiogenic factors discovered. It's produced by tumor cells and other cells as well. And there are a lot of different of these compounds that target vascular endothelial growth factor, either in a direct manner or in an indirect manner. Here is endostatin bevacizumab. Is this therapeutic, or Avastin, you've probably heard of this one before. It's a monoclonal antibody that targets vascular endothelial growth factor. And this is a multi-billion-dollar drug that, was the first FDA-approved anti-angiogenic therapeutic, and was basically the therapeutic that proved that anti-angiogenic therapy could be useful in the clinic. But again, a lot of the efforts currently, which are currently undergoing, are focused on vascular endothelial growth factor. I was just curious, and I did a search on PubMed about angiogenesis papers in general, and how many of those papers are focused on VEGF. So it turns out that angiogenesis papers keep increasing. This is the last one for last year, 2012, and we're reaching like 1,400 a year. And from all those, about 50% of those papers are dedicated to VEGF, to vascular endothelial VEGF. And if you put all of the other angiogenesis compounds combined, they're in about 10%, of the papers. So again, this is, and that's why I put here diversity. There is kind of like a lack of diversity in the angiogenesis field. There is a lot of people focused on vascular endothelial growth factor. And in my opinion, there are a lot of therapeutic opportunities that need to be explored, which will be down here on this graph. Does anti-VEGF therapy works? And the answer is yes, at least in preclinical models and also in the clinic, as I'll show before, after. But so this is just an example. This is an HM7 human colon cancer. Prior treatment with anti-VEGF therapeutics and after 48 hours after. So as you can see, 48 hours, this antibody requires 48 hours and pretty much like 90% of the vasculature is gone. Okay, and the tumor is already regressing significantly. So yes, it does work in preclinical models, and we use it all the time, and it consistently works very well to block tumor growth. Some examples of those therapeutics that I showed before. This is Avastin is not the only one. There are other therapeutics, basically, or drugs that target vascular endothelial growth factor. So what happens in the clinic? And I already was telling you before that there are numerous challenges actually to translate what we see in preclinical models into the clinic. So these are some of the successful phase 3 clinical trials with some of those drugs that I showed you in the previous slide. So there are a lot of them done with bevacizumab, either alone or in combination with all kinds of different chemotherapeutics. And as you can see, there is a number of clinical trials where there is a benefit in progression-free survival, but not in overall survival. So the take-home message for all this slide is, yes, it helps sometimes in progression-free survival, but in a lot of cases, we don't see overall survival. The other issue that is worth mentioning here is that the benefit achieved with Avastin or bevacizumab so far is often measured in weeks or in months, 2 months, 3 months for the patient. So it is a success. It was Avastin, as I said, was the first FDA approved anti-angiogenic therapeutic, and it did demonstrate in the clinic that it is effective and that it can cure patients. But if you take the average, the overall is basically the benefit is measured in weeks or in a few months. So it definitely, at least in my opinion, I think in a lot of in the opinion of a lot of people in the angiogenesis field, it didn't live up to the expectations based on the preclinical studies. There is also a lot of unsuccessful phase 3 clinical trials with bevacizumab. And again, bevacizumab and other anti-angiogenic compounds, either alone and in combination with a lot of, all kinds of different therapeutics. And so there's a lot of clinical trials in which we don't see progression-free survival or overall survival. So on top of this, there are preclinical indications, and these are 2 papers that were published in the same number of Cell, I think in 2009, suggesting that treatment or chronic treatment with therapeutics that will target vascular endothelial growth factor pathway could have deleterious effects in terms of potentiating, which is a little bit counterintuitive, but potentiating metastasis, potentiating angiogenesis, and you already said metastasis. So this is obviously concerning, and there are other publications that have corroborated these data. Whether or not this is seen in the clinic, I think is arguable, and I think is a matter of discussion at the moment. There is a bunch of publications that focused on the fact that when you stop anti-angiogenic therapy, tumors grow like crazy. So you can stop the growth of the tumors during the phase of administration. Once you stop that therapy, the tumors come back even much more aggressively than they were initially. There are other publications like this one in here, and there is a number. I mean, this is not a comprehensive list, like this one in here that talk about related mortality with bevacizumab or Avastin. So Avastin seems to have some of these deleterious, basically, or unintended consequences in the clinic. In general, all anti-angiogenic therapeutics have the problem that they are hypertensive. They cause hypertension. And this is just a problem with many or almost all anti-angiogenic therapeutics. And you'll find, this over and over again in the literature. Most of these effects are manageable. Some of these are not manageable. And in treatments with bevacizumab, you may have like about 4 times the chances of developing a pulmonary hemorrhage. You have about 4 times the chances of developing gastrointestinal hemorrhage and other complications, which are obviously not manageable. So there are problems basically with bevacizumab. So there's a lot of work being done to understand, is this a problem exclusively with bevacizumab? Is it a problem with other types of anti-angiogenic therapeutics as well? Does this, apply generally to the vast majority of patients? A slide to summarize basically the challenges with anti-angiogenic therapy. There is an obvious gap between the preclinical and the clinic. And there are modest clinical benefits. Angiogenesis is a fundamental process both for homeostasis and embryo development, and therefore is an extremely redundant process. If you block one pathway, there's another pathway that is going to take over. You block those two, there's another pathway that is going to take over. So this represents a tremendous challenge in terms of drug development. We've already talked about adverse side effects. The mechanism of action is incompletely understood. This, in my opinion, is one of the most important things. We understand a lot of what happens in preclinical models or in vitro systems. When we go to the patients, we really don't understand very well why bevacizumab is able to cure patients sometimes. We don't understand it very well. So this is one of the, in my opinion, one of the major problems right now, which is related to the last one that I write here, which is the lack of biomarkers. There are not good angiogenesis biomarkers overall across the board. There are not good imaging biomarkers. There are not predictive or prognostic biomarkers. We don't have biomarkers to stratify patients. So bevacizumab is able to cure some patients. We just don't know which patient is going to get cured. So this is one, in my opinion, one of the major problems that we have right now. What are the needs as I see them? Well, we need a better understanding of, how the anti-angiogenic therapy works in patients related to this pointing here. We need predictive and prognostic biomarkers that will help us basically stratify patients and to administer these therapeutics better. And one of the problems that bevacizumab has is that vascular endothelial growth factor is produced by tumor cells, but it's produced by inflammatory cells. It's necessary for homeostasis in our body. So it is not unthinkable that treatment with an anti-VEGF drug is going to affect basically the patient sometimes in dramatic ways. So what we need is we need targets, new targets that are specific for the tumor vasculature, as opposed to a pro-angiogenic factor that plays a role in a lot of different physiological processes. So during the years, I've been involved in a number of efforts to try to tackle these needs, and I'll show you some of those. First, are there other targets, angiogenesis targets, apart from VEGF, or apart from some of the classic pro-angiogenic compounds? So this is a study that is a bit old now, but I like, because what it shows is that, and this plot over here is showing the established microvascular genes with established microvascular function. And in this graph over here is showing other genes that they found that are related to microvascular function, but we never heard of, or that they are not the classic genes for microvascular function. And as you can see, there are actually more of these genes than in these genes in here, in this plot over here. So the answer is yes. There is plenty of targets. There is plenty of genes that are related to or that are important for angiogenesis that we're just simply not looking at. The question is, how do you find which are these genes and which are important, which of these genes are more druggable or are more interesting from a therapeutic perspective? So one of the, in my opinion, crucial moments for angiogenesis from a therapeutic perspective occurred in 2000 when Brad St. Croix, which is actually who is the PI in our group, published a paper which I think is a hallmark in therapeutic angiogenesis. He looked at what was the transcriptome in tumor microvasculature and compared that to normal vasculature, just to understand what genes, are upregulated, downregulated, differentially regulated in tumor microvasculature. And he took a very interesting approach. He actually took different approaches, but this is one of them. He used the normal vasculature in normal resting liver as normal vasculature. He used regenerating liver as an example of physiological angiogenesis. You cut a liver, the liver grows back, and that process is very much dependent on angiogenesis, and we can consider that physiological angiogenesis. And then, he had some tumor-bearing livers, which are his source for tumor vasculature. So he used SAGE analysis technology, basically to compare the transcriptome between the vasculature in all these situations, and he came up with a number of what he called tumor endothelial markers. So these are markers, these are genes that are overexpressed in tumor vasculature when we compare to normal vasculature. And he did a lot of work not only to validate, but also to understand which ones of these genes were more interesting from a therapeutic perspective. So he came up with a number of genes that are membrane receptors. So he thought that those will be more targetable than other genes. And here are some publications that talk about all these that I'm summarizing in a minute. But one of those genes is called TEM8, tumor endothelial marker 8, and he was able to develop therapeutics for this particular gene. This gene is expressed. He demonstrated that it's expressed exclusively in the tumor vasculature. He did analysis of the expression of TEM8 in vasculature in a panel of different normal tissues, and who could never find it there, exclusively in the tumor vasculature. And what was interesting is that he also developed a knockout for this particular gene. And the knockout shows absolutely no phenotype, except for a defect in the teeth that are misaligned. So the knockouts are perfectly viable and normal. And however, when you grow a tumor in these mice, the tumor growth is greatly delayed. So this will be the growth of the tumor in the wild type, and this is the growth of the tumor in the knockout littermates. So this result is not so surprising, and it's not surprising because what he was looking at, this tumor endothelial marker 8, is only exclusively overexpressed in the tumors. And therefore, it's not surprising that if it doesn't play a role in normal physiology, these animals are perfectly normal. And you can only see the effects when in the context of tumor growth. He went ahead and developed antibodies to target this particular tumor endothelial marker 8. The antibody is called L2, and as you can see, when he treats the animals with his antibody, he observes a similar response to what happens with a knockout animal, or even a similar degree of inhibition, of tumor growth inhibition, suggesting that he's able basically to block pretty much all the TEM8 present overexpressed within the tumor. And he went ahead also and did a lot of different combinations with other chemotherapeutics and show that TEM8 synergistically acts with other either chemotherapeutics or other anti-angiogenic compounds to suppress tumor growth. And he was able to achieve also tumor-free animals in some of the studies that he did. one experiment that I wish I would have added in here is, which is I think is beautiful, is in the same animal, he put in one side one tumor, and in the other side of the animal he made a wound. Okay, wound healing is very much dependent on angiogenesis, and he went ahead and treated that animal with this antibody, which is again a monoclonal antibody for TEM8. What happened is he was able to regress the tumor in that animal, but the wound healed at the same speed that in animals that were untreated. Again, one functional basically example that the antibody is basically having an effect on normal vascular, in tumor vasculature, but not in normal vasculature. What other approaches can we take to devise novel targets to target specifically the tumor microenvironment? Well, in the process of drug development, there is always pretty much a process of high-throughput screen of some sort. And in the previous presentation, she was talking about these massive screens with RNAi. In the past, I tried to devise a way to come up with an in vitro assay that was meaningful from the perspective of tumor angiogenesis that could be used to screen large numbers of drugs. And this is the A2 formation assay. I'm not sure if you're familiar with this assay, but it simply consists on generating a three-dimensional matrix of a gel that is composed of an extracellular matrix from a tumor mouse, from a mouse tumor. So from this mouse tumor, we extract the extracellular matrix and generate these 3D gels. And on top of those gels, we add endothelial cells. So the endothelial cells are basically on their environment, on their matrix. And this is basically what happens when you do that. Okay, there you go. So the cells, which initially are homogeneously distributed in the matrix, will rearrange to form those cords that you see in there that mimic the vasculature in the tumor microenvironment. So this is what is called a two-formation assay. We didn't invent it. It was, it's been around for a long time. But I thought that we could use this assay to do screens, basically, of drugs. To make a long story short, this is just an example of some of those screens that we did. And here on the left, you have an assay where we didn't treat these compounds, these cells, and that's not going to work, but basically it was doing exactly the same thing as in the previous image. And this is one of the compounds that we were able to found. This is one of those assays treated with one of these compounds. Basically, the cells are able to move around freely in the matrix, so it's not blocking migration, it's not blocking viability of the cells, but it is blocking the ability of those cells to form tubes. Okay, so we went ahead and tested these compounds in preclinical models, and this is one of those compounds that I showed you before. And actually, in this particular experiment, we were testing two of these compounds. This is the growth of the normal tumor, and this is the growth down here of two of these compounds compared with Avastin, which is we've already talked this anti-VEGF monoclonal antibody. So as you can see, we were able to find, actually, as a matter of fact, these were the two compounds that we found in these screens. We just picked two. We did the whole screen, we picked two compounds, and these are the first two compounds that we tested in vivo. So indicating that the assay is basically very robust in terms of finding small molecules. In this case, these were small molecules that were effective in preclinical models of cancer. So these are just two examples of how you can go about using unbiased systems to find drugs that are specific, more specific for the tumor microenvironment that can be potentially translated into the clinic. And I did promise that I will finish, and it's 10:06, and I will take any questions that you might have at this point. Yes?
How does M8 compare with PSMA? Well, PSMA is a very, 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 very,
In terms of what? As far as I know, we haven't done any direct head-to-head comparison of both of them. I can tell you therapeutics for TMAID have absolutely no toxicity whatsoever. One of the challenges with these targets is to really find whether or not they're expressed anywhere else in the body. There might be an obscure place, a cell type that we didn't look at, that is expressed in TMAID and where TMAID is functional and is necessary for homeostasis. And this is why all these experiments, doing, like, for instance, this experiment that I like so much, where we do the wound and we do the tumor in the same animal and treat the animal, really demonstrate that that the animals are not affected, or the therapeutics for TMAID are not toxic in this animal. There's an interesting paper that a collaborator of ours published last year, which found this group of patients that have Gappo syndrome. Gappo syndrome is an obscure syndrome that produces some growth retardation, alopecia, certain deformities, which seem to be caused by accumulation of extracellular matrix. But these people are perfectly normal otherwise. They live normal lives otherwise. So these people have mutations on the TMAID gene. It's completely knocked out. They don't have TMAID at all, right? And these people are relatively normal, apart from this accumulation of extracellular matrix. Interestingly, they have defect on the teeth, the same as the mice, the knockout mice. But that is a good indication that TMAID is not required for normal development and for homeostasis in adults. So it's a good indication that therapeutics for TMAID won't show basically any toxicity in patients. Yes?
Is that non-requirement for a gene clearly evolved pattern? Are there just redundant family members that are upregulated, or what is TMAID actually?
That's a very good question, and we're working really hard to understand that question. So TMAID actually happens to, you might have noticed in my slides that I also put another name in there. That is called anthrax receptor 1. So it turns out that TMAID is a receptor for anthrax. Obviously, TMAID did not evolve to be an anthrax receptor. And we do think that it has a function in normal physiology that is probably redundant to other genes. And so knocking down basically of TMAID is not required for it.
But its expression, you don't see a certain expression of disease, neovascularization of normal disease today?
So one of the very interesting experiments that Brad did a long time ago was he looked at normal places where physiological angiogenesis occurs, like, for instance, the corpus luteum, right? He found absolutely not expression of TMAID. And this is why TMAID is so attractive, to us, because it's really expressed in tumor vasculature, and we can't find it anywhere else.
And if you induce expression, then you cause more logical conditions.
If you induce forced expression in a transgenic, we haven't done that, but yeah, we haven't done it. Well, we can overexpress TMAID, and of course we have a lot of different systems where we have overexpressed TMAID in vitro, and the cells don't seem to show any phenotype in terms of growth or morphology, any of those overt things that you will be able to see.
And is the reason for sticking with people with targets that that's a druggable target, or because what's the terms of $10 billion on that one target?
Well, this is kind of like in my opinion, is kind of like a historical reason. This is an entire field in the search for one target that we can translate to the clinic. So Genentech and Napoleon Ferrara made this antibody for VEGF, and it was the first FDA-approved drug, and they showed that it can work in the clinic, and that attracted massive interest into VEGF. So that has done a lot of good for the field in terms of we've shown that we can translate these things in the clinic, and it's effective. But at the same time, it's probably not the best target because of all these things that we have discussed today, right? But it's probably a historical reason from my perspective. I think you wanted to ask a question.
So with your xenograft, across the assay xenograft, are you looking at mechanical sequence? Would you expect it to have like the endostat results?
The endostatin results are very hard to reproduce. But yes, we are moving, or we're trying to move therapeutics, in particular monoclonal antibodies for TMA, to the clinic. Yes. More questions? Thank you very much.